The sabbatical idea really started in fall 2017, I think. Is that right? Yeah. So we applied for it, and the idea was to take a look at the amount of mercury in predatory fish in the Des Moines River or in Des Moines, Iowa, or not Des Moines, Iowa, Des Moines rivers. And so what we settled on, we're looking at catfish. And so there's kind of two things at play, and the terms that we would use would be bioaccumulation and bioamplification. And so the distinction between those two is that in bioamplification, as you move up the food chain, environmental contaminants generally concentrate. Bioaccumulation is when you look at one individual in that species. If you look at it over a period of time, when it's a young fish, it hasn't accumulated a lot of contaminants yet. As it gets older and older and older, then the contamination builds up. And so we were interested in the bioaccumulation in an individual. So generally speaking, the idea would be, let's try to add some clarity to the recommendations that the DNR talks about in terms of consumption of fish. And so can we say catfish above 28 inches or 30 inches or some kind of threshold are no longer safe to eat. And so we started that collecting in the summer of 17, carried that into the summer of 18. I don't know how many fish we caught, probably close to 200 fish. We didn't sample all of those. One of the things that we also wanted to do was try to model, put together a mathematical model of the accumulation of mercury in a fish. And so part of that was to try and figure out how fast fish grow. And so some of the fish that we caught, we actually tagged and then released back into the river. So we caught a bunch of fish. We attempted to analyze the fish in-house, but the levels that we needed to look at would be 50 parts per billion, so about 200 times lower than what we were actually able to see in our standards. And so we ended up sending our fish samples to the State Hygienic Lab up in Ankeny, and then one of the students that I have working with me, Alex Austin, was able to fit that data to the model that he created. When we looked at the model, what we determined is that it has a lot to do with the concentration of the fish, bait fish, that they happen to be consuming. One of the interesting outcomes of this is, we didn't necessarily know it, but the DNR was also carrying on a study that was very, very similar. Instead of just looking at flathead catfish or catfish in general, they looked at a variety of different species in Iowa. And so we've gotten to the point now where we contacted the DNR just to ask some questions, and that led to a conversation where they told us about what they were doing, and we asked if there was some way that we could work together and help. And so one of the interesting outcomes that I think, at least, in sort of a good outcome for our students, is that it looks like in the future we're going to be able to collaborate with the DNR, maybe get some of our students out into the field with their research agents. The other nice outcome for us is the research student that I had most recently, Alex, presented his work at the Regional American Chemical Society meeting up in Ames in October, and then him and I are both going to go to the National American Chemical Society meeting this spring, which in the 9 years that I've been here, I don't think we've sent a student to one of those meetings, and so it's kind of neat to do some work with students and then have them make enough progress to be able to put together a poster in his case and go down to a national meeting where there's thousands of posters being presented and tell people what he did. So.